Hello, this is Group 14, and we will be discussing the pyruvate dehydrogenase multi-enzyme complex, or PDC. Specifically, we will be discussing its organization, its mechanisms, and its susceptibility to arsenite, a toxin that can halt the function of PDC.
Oftentimes, enzyme-enhanced reaction rates are limited due to various factors, such as side chains occurring between intermediates that can hinder the reaction rate by removing valuable intermediates from a reaction sequence or otherwise affecting reactant product equilibriums. Additionally, multi-enzyme reactions require several points of regulation and are hindered by intermediate diffusion rates between enzymes. One process with interesting solutions to these problems is the pyruvate dehydrogenase complex, which consists of three enzymes that each fulfill a role in the development of acetyl-CoA. One important component of the PDC is its multi-enzyme complex structure, with the structure being crucial to the rapid and efficient conversion of pyruvate into acetyl-CoA within the mitochondrial matrix. Three primary advantages exist for placing multiple enzymes in a complex. First, close enzyme proximity reduces diffusion times of intermediates between enzymes. Second, channeling of intermediates between enzymes minimizes side reactions. Finally, enzymes in a complex can be regulated at a single point, making all reactions capable of coordinated control.
The PDC has three enzymes alongside several coenzymes and prosthetic groups, which undergo 5 reaction steps. First, within E1, or pyruvate dehydrogenase, a carbanion in thiamine pyrophosphate, or TPP, performs a nucleophilic attack on the carbon of pyruvate, forming a tetrahedral intermediate complex between TPP and pyruvate. Pyruvate is in orange-red and TPP is in blue. Then, pyruvate is decarboxylated, resulting in the release of CO2 in hydroethyl TPP complex group attached to E1. Hydroethyl TPP E1 is transferred to meet the lipoamide of E2, or dihydrolipoyl transaxylase. Next, in a nucleophilic attack of the hydroethyl carbanion of the lipoamide, disulfide bond results in the formation of acetyl dihydrolipoamide E2 and an E1 form with the regeneration of catalytic TPP. Following this, the acetyl group of acetyl dihydrolipoamide E2 is nucleophilic attacked by the CoA sulfur, inducing a transesterification to form acetyl-CoA and dihydrolipoamide E2.
The next part of the reaction involves the regeneration of E2 and the transfer of electrons to the eventual destination of NAD+. E3, or dihydrolipoyl dehydrogenase, is made up of an FAD core, highlighted in blue, as well as an intramolecular disulfide bond. It uses this disulfide bond as an oxidizing agent to reform the E2 lipoamide complex, leaving reduced E3 that contains two sulfhydryl groups. FAD then acts as an oxidizing agent to reform the disulfide bond of E3, leaving a bound FADH2. This FADH2 is reoxidized by NAD+, leaving catalytic E3, NADH, and a proton.
One advantage discussed with the PDC is the ability to channel metabolic intermediates between enzymes. But how does this work? The structure of the R group on E2, shown in this figure, has a fully extended length of 14 angstroms. Here, the catalytic sulfurs are highlighted in yellow while the carbon arm is green. This lipolysyl arm, in conjunction with a 140 angstrom outer linker group mentioned by a different group, can fully extend and move the attached lipo group between the active sites of E1, E2, and E3. Picture a monkey swinging through a treetop, carrying a banana from one tree to another. Similarly, these lipolysyl arms can interact with each other as well, exchanging acyl groups and disulfides with each other, as shown here. In this way, a single E1 or E3 subunit is capable of acting on multiple E2 subunits through the functionality of the lipolysyl arm, and the enzymes are no longer rate limited by one another through the channeling of substrates.
Arsenic compounds such as arsenite are toxic to the body as they can disable the PDC. Specifically, they covalently bind to the sulfur molecules on dihydrolipoyl dehydrogenase, or E3, of the pyruvate dehydrogenase complex, forming a bidentate adduct between the arsenic and the reduced sulfurs on E3 while producing water. This arsenic E3 complex can't be oxidized by NAD+ to its catalytically active state, causing the PDC to shut down, as E2 can no longer be oxidized by E3. Microorganism PDC enzymes show differences in sensitivity to arsenic compounds when compared to human PDCs, which paved the way for organic arsenic compound use as the first antibiotics. These antibiotics were used to treat syphilis and trypanosomiasis, but had severe side effects. Nowadays, organic arsenic used in syphilis treatment is superseded by penicillin to reduce the toxic side effects.